Okay. It's still morning, I think, so good morning. My name is Talia Karim. I think I know a lot of you guys. And thank you for coming to our session, along with our other moderators, Erica Kermel and Holly Little. We'll be keeping time and introducing speakers today. So welcome. I have to read a little thing. Okay. This session, entitled Spinach to Tadwig, Bridging Communities and Building Capacity for Digital Collections Mobilization. I already introduced myself. I'm Talia again. I am at University of Colorado. I manage the invert paleo collections there. This meeting was made possible in part by the generosity of our sponsors, especially our diamond sponsors. We are grateful to them and to Oki Congress for today's tech support. So thank you guys very much. The meeting has been wonderful so far. This session will be recorded for later viewing by registrants and eventually posted to the Spinach and Tadwig YouTube channels for public viewing. Please speak slowly and clearly for the benefit of all attendees. We had a lot of folks yesterday on Zoom. And thank you for joining us. So we will hopefully have time at the end of each talk for questions. And if not, we have a little bit of time at the end of the session for discussion. So stick around if you have thoughts you want to share about building capacity for digital collections mobilization. And with that, I will hand it over to our first speaker, who is Deb Paul.
Hi, everybody. It's very exciting to be here and see familiar faces and new faces and see the folks online. Hello. I am currently, some of you know me from iDig Bio, and now in my current role for the last 3 years, work for the Species File Group as Biodiversity Informatics Community Liaison, bridging the distance or the gaps potentially between those who use the products and ask for things and those who build the things that we need in order to do the things we want to do. So I'm here to share my experience and our group's experience about things we're learning about this human and data ecosystem change. So from a story point of view, we are a system of networks. Sometimes they're connected, sometimes they're not. Who does the connecting between those networks? So we are communities that are also working to connect open data, right? And so from that perspective, we have known issues we've discovered and unknown issues are unknown to us. So we need to make opportunities to share those things, like this symposium. And we need strategic ways to share so that we can propagate what we're learning and we can continue that conversation. And we need ways to evaluate the frameworks that we actually have in place. Are they working for us? So I have a question for you. Think of a collaboration. It could be at home, in your own home. It could be in your job. What's working and what's not? What are some things about different collaborations that you have found, man, this was really great because? What comes to your mind? So just make a mental note. Or what's not working? Why? See if some of this resonates with you. So I found this model. Some of you may know Elinor Ostrom. Some of you may have heard of the tragedy of the commons. We have a common pool, right? We are a human common pool. We have limitations, right? We have not enough people. That's been a common theme. And in addition to that, we have limited resources, and our planet itself is a fixed resource. So are our systems working for us? How do we figure that out? And how do we use what we learn to make them better using this model, which Elinor Ostrom won a Nobel Prize for, to address the conclusion of the tragedy of commons that the only way to fix it was for governments to dictate that people couldn't collaborate to do things together. And she said that's not true. So there are some key attributes. There are more than are listed here. I've selected this notion of how do we form strategic alliances? Who gets to sit at the table? Are we providing benefits to diverse groups in the pool or just people at the end of the pipeline? Are we building processes that foster learning or dependency? And are we having success in overcoming our shared challenges, which we've heard many of this week already? Think about that in terms of the question I posed to you just a moment ago. So where are we having some success from a local perspective? My local could be in a group, it could be our Species File group. We are noticing that awareness, as the symposium organizers point out, is key. So once we make our groups or people that we interact with aware of opportunities, aware of software or networks, this begins to change their ideas about what's possible. They become exposed to, quote, new tools. They become part of development of new workflows and new collaborations. They adopt new ways to do things, and then they begin to ask new questions and make demands for new things and build their own connections. So they become a functional part of the network itself. So whose job is it to connect the networks? Whose job is it to do this liaison and biocurator work? And how do we embed these people, add these people, and give them credit for the work that they're doing? They're not typically credited in academic institutions. This work isn't, there's no checkbox for it. So where are some places we've had our door gets knocked on? External in the sense that we're not doing the work these people are doing. For example, in the Illinois Natural History Survey, we're facilitating what they do. Here's the things they ask us. Can you help us write a data management plan? How do we publish our data set? Where do we publish it? How do I write a data paper? I didn't know there was such a thing, right? How do I deal with people identifiers for our institution and why are they valuable? And we build bridges. We've now made possible for those of you who use iNaturalist, if you find an orthoptera name and it has a problem, you no longer complain to iNaturalist. You click a button in iNaturalist, but it goes to the Orthoptera species file project, housed in TaxonWorks, and you tell them about it and it gets fixed. So agency. We've exposed all these people to, quote, new tools, empowering them to do things they didn't know were possible before. We include building that into our software, and we are trying to move people toward new ways to communicate with each other and build their own communities. So where are we having challenges that we see need continued work? These are very broad and they're at very different levels in the local sense. People are asking for help. This is a win. How do I move beyond email? How do we have more transparent communication for our group or our network? They are asking for, how about credit for software development? This is not a thing in academia. How do we engage with communities who've been burned? This is something we've seen by infrastructures or projects that have died. They've had their 5 or 10-year funding and they go away and the communities don't want to engage again. We see a better data need from the start, better map to standards, more shareable, more fair from the beginning. We can only do so much to put Band-Aids on it later. And we see a leadership awareness issue that could use some work I'll talk more about. We can speak to ourselves here and go, oh yeah, we need that, but the people in the room who can give us that are not in this room. So keeping this polycentric governance model in mind, here are some ideas. So one is this Paleo Data Working Group that has organized this session gives us a potential model for addressing some ways to go to other discipline groups and do the similar kind of work for things like helping those groups build more effective controlled vocabularies. We have groups like the NFDI for Biodiversity project that is a German research infrastructure project. They link 15 to 20 different resources together. A unique thing they did was build a data help desk that's integrated. So if you go there as a researcher for help, you don't have to go to each individual resource for help. They've integrated their help desk so you don't have to figure out how to get your data from one to the other transformed. You can get your help all in one place. They've also written into their funding a way to support those infrastructures if they should lose their financial footing or their infrastructure for housing data, for example. Other possible ideas are to continue these in a more strategic way, these conversations we've been having about collection management software, about controlled vocabularies, this polycentric governance model. How can we look at a higher level of what we're doing and what's working and what's not? And can we agree that there's a need for local capacity development, local credit needs, professional development? I see that as a shared need. How can we be more strategic instead of just saying that we need it? So one thing I've noticed recently is when I have gone to some discipline-specific meetings recently, there are individuals giving talks about what's interesting to them. What's their lab working on? What are their current research results? There's very little strategic conversations going on about biodiversity informatics needs. Is my data working for me? Can I share it with others? Is my data fair? How do I do that? Those conversations are not taking place. They're not present in the program anywhere. And I see that as an opportunity for both spinach and Tadwig. And I'm curious to see if that resonates with you. If you see at your discipline-specific meetings, is this something you see missing? So on that note, I have many. This is 3 that I limited myself to. Ideas for you, but I'd love to hear yours as well, is moving forward to strengthen the connection between spinach and Tadwig. And one would be that we have a memorandum of understanding. It essentially is like a reporting strategy where we just sort of tell each other what happened each year. I would like to see us add things like standing sessions at both meetings. So when you go to a spinach meeting, there'd be a session that was like Tadwig based, that was where the intersection conversations can happen. And the same thing at spinach. I'd like to see something I and others have talked about in the past. Collection managers, how many collection managers are in here? And how many of you would say you found it extremely valuable when you visit another collection? Yeah. So instead of making that the lucky few who get to come to spinach, the lucky few who get to visit the collections, why do we make that the luck of those of you who have the money or the wherewithal to get to those meetings? Let's figure out a way to make that part of professional development where you can go to another collection and spend a week there or whatever, and vice versa, those can come to you. This is another way we can foster culture change. We can, and share, what we're all learning. And the last one I would say is that we have an opportunity to address what I see at these discipline-specific meetings together. Thank you. And as part of that, I'll also work on this leadership awareness need. So we can all agree that we need these things, but if the leadership at these discipline-specific meetings doesn't get that, then we're just talking to ourselves. So we need to figure out a way to talk to them. And one concrete example, since I have time, is at the Ecological Society of America for several years in a row. And I realize now again, IDIG-Bio and others have been building these data help desk events where we're in the exhibitor space and we have multiple organizations presenting this. And one year, the Ecological Society of America came to us and said, we provide career development track at our meeting. How to write a better resume, how to. So they said, you guys have great content for professional development for ecologists. Can we take what you're doing and stick it under our umbrella of professional development? And we said, yes. It gets us a space in the program, it gets us more vision. But they're the only ones so far that have asked for that kind of thing. But it shows that there's a recognition there that they need that in their community. A leadership awareness, that's what I'm saying. So with that, what are your thoughts? What's working in your models? What's not working around these benefits to diverse groups in the pool, processes that foster social learning? Are we having success in overcoming linked challenges, and what are those? Thank you.
Anybody have questions for Deb? We also have discussion at the end.
Thanks, Deb. That was a really nice talk.
Thanks, Nikki.
I see we often talk about how we bring more people in, and I think we don't very well manage the converse, which is how do we let people step back from their responsibilities? And I worry about that a lot in the software tools and the expertise that we have in this community. Mm-hmm. So have you got any thoughts about how we
Can you give me a concrete example?
So we explain how we can bring people to projects.
Right.
But I see a lot of projects, some of the BHL technology that say the things that Rod develops, mm-hmm. They're often single developer things.
Sure. What
do we do when those people stop? How can we encourage people to design their things in such a way that they can step back sustainably? Yeah,
what? I think, Nikki, that's a big conversation about culture change in general. To me, that's the same thing as a taxonomist working for many years, and then when they realize they're getting ready to step away from their formal career, they say, oh my God, here's all my data that only I understand, and there's no metadata, and I didn't put it in a shareable place, and it's not fair, and nobody else is going to understand it because of me, because I didn't write down any metadata, and can you help me, like now? And I think it's the same kind of conversation in the sense that how do we build systems up front that put that expectation that you just expressed in place to start with?
That is a great question, and let's file that away for discussion at the end, because I think we could talk for another. We could talk for a long time on that one. Thanks, Deb.
Thank you.
Okay. All right, our next speaker is Jessie Nakano. Come on up.
Hi, everybody. My name is Jessie Nakano. I am an assistant registrar in the Department of Paleobiology at the Smithsonian Institution. Our presentation today is the Unifying Community Efforts on Registration and Data Rights, and that is in collaboration with Holly Little, who is my colleague, also one of the chairs today. She's in the front, and she is our informatics manager for the Department of Paleobiology at the Smithsonian. Our presentation today will highlight notable outcomes from almost 4 years of our joint work at our institution. These identify key linkages between collection transactions and the digitization of paleo collections at the Smithsonian. These finds were primarily possible due to the community engagement outside of our immediate kind of institution and even our department. And it's an emphasis on this kind of the value of a multi-professional expertise. And we think it's kind of really a good discussion that we wanted to share else. We also will highlight though challenges and some ongoing issues that we have. And I think it's really good for us to have this discussion with everybody, especially with the community here, but also start to engage elsewhere and kind of identify kind of what a lot of things Deb talked about. So I will jump in. To kind of start, we have a recap kind of the work we've been doing. It's kind of a summary starting back in 2020 when Holly and I really started engaging a bit more about each of our separate roles as a registrar for the department and as the informatics manager for the paleo department. We really started to kind of we were asking our own kind of similar questions and conversations about the work we do and how that's impacting the paleo collections. So Holly was really involved a lot with the data side, obviously, the responsibilities of care, mobilizing the data, the life cycle of it. And my role was really into the registration about documentation, acquires, the loans, and then those rights associated and how that is a long-term managed. And it kind of started to see how we can handle data really depends on how we get a physical specimen. So that kind of really was what started back in 2020, and we started having conversations separately and together. So this timeline kind of I see it's getting cut off, but ignore it. But it kind of gives you a good capsule of like the different groups we started engaging. So this is our information gathering, seeing internally, externally, at larger levels, and in different capacities, individually and together. With that, we found that there is a very intricate, interlayered communities within overall across this topic. We really started to see the scale and how we see us we scaled it down. We're in the middle. We have our paleo department specifically, and then kind of working outwards to each level. And that kind of was a little overwhelming in the beginning because you started to see like there's this through line each, and we have one idea that would then take us all the way out, but then pull us back in because we're like, how do we address this at our level, but at these other levels, and then different challenges too and obstacles each way. So it kind of gets to start off, we're going to start in that little dot in the center. And so we're going to talk mostly about the work that we did for our specific department. This really was what we kind of came to realize was like setting a foundation and a base for us, even if it's not the base and it's going to stay like that forever, at least it sets a foundation so we feel comfortable to implement and then kind of learn from there and build and readjust based on our communications as we kind of go through each of the layers in engagement with different communities and expertise. So as you see here, so one of the things we found really unique for us within our department is our role specifically. So you having a dedicated registrar and then also having a dedicated informatics manager is very unique even for our institution itself. And that's something we started to see, like how do we utilize and show the importance and value of, even for like registration itself, we often are lumped in with collections managers, or it's a collections manager holding multiple hats. And we realized like you really need to start thinking about if you're putting too much effort and all this responsibility on one person who's not necessarily has to be their skill set, and you can have an expert who's really focused on working on that. So for us, we revised and made shareable department workflows kind of based on registration needs and like what that value is and what that workflow would be, the data entry and overall collection services. One thing that we did start to use in terms of thinking future forward and kind of thinking more at a global scale and like landscape and the global landscape in mind was setting up our resource accounts. So while it's right now managed by individual people, so like Holly has one and I have one, each separately for our informatics and registration, it's a resource account that will have a legacy and could exist within our department, our institution after we're there. So there's a sense of like records and history that is referenceable to see those decisions, even if it's a correspondence, but it gives us that kind of baseline. Overall, we also started to kind of figure out better credit lines, loaning of specimens and that process for us, and specifically to a decision tree. So this cycle here kind of emphasized how our process was and just making it very clear of like what stage collections management in general, including informatics and registration, registrars should be involved to make sure that that's assessed before something is fulfilled by our researchers or approved beyond, because that tended to be the little hiccup where that bottom tag was kind of jumping up everywhere. So then we had to do a retroactive and figure out what was happening. So, you know, it's fun. Also, as a registrar, I really like documentation in words, so you're going to see a lot of pictures, but there'll be a lot of words. But either way, there's a QR code. This is our website that we started really focusing on these past year to kind of just get some language out there, even if it's very simple and basic. It at least gives you a contact, a resource account, but a better idea of like how we handle data of our digital assets and data rights for our specimens that we're scanning or taking images of, and including how we want to cite our specimens. And like, ours is very complicated because our acronym is USNM, but we are also referred NMNH and Smithsonian. So there's a very wide gap. So trying to just make it clear of like how it is, there's a lot of prefix things, but this link should give you a link to our website. So the next layer you see there kind of goes into the next level is our Smithsonian as an institution, and then our unit as a museum of the Natural History Museum, which is one among many kind of more art and cultural history, cultural heritage museums. This was kind of a really important step for us because even our museum itself is not as coordinated. So one of the things that we kind of did back on that timeline was set up a specific group within our institution, within our museum that was specific to intellectual property rights and open access. And our Smithsonian open access policy was kind of the driving force about it to kind of get us all on the same page about what those rights are for our collections, but also trying to engage with better on kind of what we could do as a whole instead of individually as departments, because each of our departments and even divisions sometimes operate very individually and separate. So what they do, we weren't even communicating. So like getting that all into one, as close as we could into one. And these are the examples of different agreements and forms we've kind of worked to standardize. And then the biggest thing we had to was updating our collections management policy to specifically highlight physical collections, but also the digital collection side. So born digital, digital assets, and those faithful reproductions and what that really means. So it was very clear that we understand that those are part of our collection management policy and those are important for us. And this is an example of our two different terms that we have to rotate between for usage rights for digital assets. So we have our usage conditions apply, which is a very standard, like it's restricted, and you need to request permission. And then the CC0 is our open access. So there's fair use for anything you'd like. The next layer kind of was the discipline and formal groups. So this is kind of taking us outside of our institution, but then making us a little bit more focused on other paleo collections. You know, like who else has paleo collections? What are your problems and your issues when it comes to data rights, but also a little bit more of like, what are the issues you're handling and who are your experts handling it, but who are you don't have that you don't really have the time to consult with? So like, can you talk to another institution and they might have someone who can help provide what they're doing and give you insight. For one of the things we did and kind of presented within that smaller group, but since it's informal, is kind of a good test example for us to kind of like push out ideas and things that we were trying to create. So one of them was our transfer and release agreement, specifically about fossil casts and those, and also scanning and 3D prints, but trying to make a little subsections about the digital data, because for paleo collections, I don't know if others have this, casts, people are really trying to scan, CT scan casts as well. So trying to make sure we realize that on a global scale conversations that is happening and people are utilizing it. So how do we make sure that that communication is clear and continuing to go? This is kind of our good example of like a project where, and Holly's in this picture, if you can find her, back kind of in 2012, 2013, when the Smithsonian Digitization Program Office first started, and kind of did this scanning of a site with one of our curators, researchers. But then trying from there, we don't necessarily own the physical, but we manage the data. And then there are 3D prints that exist and those permissions associated. So this is from our 3D collections online website. And you can see that it has the physical owners of the physical specimens catalog numbers. So it's a little bit more specific there. So you can see that it's like clearly identifying it that way. But then how do we know 3D prints and then where that data and what you can do with it? Can you scale it up or down? And what does that mean for people to be more communicating on it? The last big overall is the global engagement. This is kind of one of the last outer thing, but something that I think we're really realizing what we need more of it overall. There's two separate groups. We have Tadwig and Spinach here at this conference, and so it's really important to kind of have those communities merge and start to kind of think about how can we work together on these ideas, but also have our expertise be utilized in a better way. For us, it's like really, there's a level of knowledge that someone or one person needs, and you can't really rely on that person to have all the knowledge. So it's like, how do we start to think about it better of like, this person is this expert and I'm going to rely on them, but like they can be focused on, and if it's too much or too little, like expanding and where it kind of capacity goes. And how we do that on a global scale as well, and having those conversations of other institutions lay out or formatting with staffing, but also their engagement as well. For me, I kind of really started to see from a registration standpoint, like we often are lumped in and there hasn't been sessions ever focused specifically on registration. So I've been trying to really push and try and get a better group that really has anyone who's identified or has a role as a registrar versus everyone who has, who wears multiple hats and can do that work. Yeah, this is more of the pushing off point into our ongoing challenges. So it continues to be, and that's where the registrar comes in, where the balancing and compliance with policies, laws, and regulations. We still have issues in terms of tracking and recording digital assets in our CMS. Like, how does that go? How do we make sure we identify it versus adding it in there? Another thing is scientific illustrations or archive collections, those digitization of it. That's kind of a big red flag for us, and we've been thinking like maybe we need an archivist or talking to library science who have that expertise, so we don't try and learn something new and figure it out, and when we don't need to, if there are already people who have that knowledge and can help share and provide it to us. So this is my question for everybody here, and also just to help continue this conversation afterwards. But how do we better kind of engage the community to address overlapping data standards and registration needs? Are there other roles that we should really be thinking out and promoting as roles within our institutions and managing our collections and the data and the digital assets of them? Are there gaps and other obstacles, and kind of how can we have a conversation that is effective and more overall engaging? But yeah. Thank you. That was about it. I'll leave that there. Oh yeah, and that's my contact, so you can find me.
I'm gonna go back to Jesse's other slide. So take a picture of this. We have discussion at the end, and we can come back to these questions. Unless anybody has a quick comment. Is there anything on Zoom? No, nothing on Zoom? Okay. Good, and then Jesse's contact information. I love this Paleo registrar email so that you have a institutional history of correspondence. That's really great. Yeah, that's part of for Nikki's question. Yeah, that gets at your question. This what did you call it? A resource email? Yeah, so that you have an institutional history of correspondence, not just like in your personal institutional email. Yeah, I like that a lot. Okay, I will invite our next speaker up, Iru Cheng.
Good morning, everyone. Thanks for coming to my talk. My name is Yiru Chen. I'm from Academia Sinica in Taiwan. So before I start, I would like to do a little bit of survey. Raise your hand if you know there's bird egg collection in your museums or institution. Oh, great. There are a lot. You're my target. So I'm a scientist. I'm interested in why there's such a variety of shapes, colors, and patterns in bird eggs. As you can see here, we have eggs that have just pure white, and we have eggs that have brown colors, and some have blue colors, some have spots, some don't have spots. And so, but we don't really know why there's such a variation. And one of the reasons is that because it's really difficult to collect egg samples. Take my research as an example. I want to understand how the environmental conditions affect the evolution of egg colors. Specifically, I am investigating how temperature and UV light along the latitudes and elevation affect the evolution of egg colors. I would love to travel around the world to collect the egg samples, but I think it's very unlikely. So I thought I would jump into the internet world and see what I can find out and if there are any digital specimens that are available for my research. So based on Bambarini's review in 2020 and some collections that I can find on iDigBio, there are in total 305 museums that have bird egg collections. I made a map, so you can scan the QR code if you're interested in if there are any egg collections near your place. So that amounts to, in total, 2 million egg sets. However, I think it is still an estimated numbers because as you can see on the map, most of the museums are in the West. But as far as I know, there are some museums in like museums of Kenya or some museums in Asia that are not covered here. However, when I dug into the digital specimens, I found the protocols, digitization protocols, are almost as diverse as the eggs themselves. As you can see here, some photos are taken with the egg still in the case. Some are taken out and positioned separately. And the background are kind of ranges over the place. Some of them have the cotton as background. Some has the black fabric. Some use the gray cardboard, and some use the carpet. Some include a scale, some don't. Some better ones have the color chart, but somehow they seem to use whatever is available on hand. The better ones, they have, like the Yale Peabody Museum, they have the standard color chart, which actually includes a standard grayscale. Unfortunately, that becomes a problem because these digital photos are not taken in a standard format, so they are less useful for scientific research. In 2017, a Spanish engineer developed a tool called SpotEgg. This is a fantastic tool that can extract the features of digital specimens. It can automatically process a folder of all the photos within minutes. It can automatically detect the region of eggs and measure the size of the eggs, also measure the RGB reflectance, which then can be used to calculate some of the variables of interest for scientists, such as brightness and blue-green color chroma. Most amazingly, it can even measure the spots on the shell, including the size of spots, the reflectance of spots of different colors, and also the coordinates, the spatial distribution on the shell. However, all this magic can only happen if the photos are taken in a standard format, which means that the background has to be in a big contrast so the computer can see the eggs. Also, it has to include a standard color chart, which has the grayscale, so the computer can calibrate the photos that has different lighting conditions that are used by different museums. So now we have inherited this such incredible egg collection assets from 100 years ago. So I'm kind of curious, like, so how far have we come in making these collections accessible to a broader community, and also what's the status of digitization of these collections? So we, to do this, we examine like the museum collections that have more than 500 egg sets and apply the FAIR principles. I know most of you are very familiar with FAIR principles, but let me just walk you through how we apply the FAIR principles to the bird egg collections. So for the collections to be findable, they need to have a digital archive. The better ones, if they are will be more findable if they are uploaded to a regional or global open access platform, such as Arctos, IDIC-Bio, Atlas of Living Australia. But for the specimens to be accessible, they need to be digitized and then also provide downloadable photos better to be high quality. However, even when the specimens are accessible, different digital specimens from different museums may not be interoperable when they are not taken in the standard SYM formats. Also, for the specimens to be reusable, some of the metadata provided associated with the specimens are also important. For example, the coordinates through the georeferencing, because that will provide scientists to use the coordinates to extract some environmental variables that's associated with the specific specimens, and that will allow us to ask more questions. So how fair are the bird egg collections? Here on the left are the percentage based on 212 museums, and the bar chart on the right are the percentage based on the 2 million egg sets. For the digital archive, about like 40, I can find 48 museums that has the digital archive. I'm more optimistic because during my journey, I found some of the egg collections are actually very hard to find and dig into it. I will explain it later. And for this, the data digital archive that's also on a non-local platform, there's about like 36 collections I can find. So about like 17% of the museums. And in terms of like the digital specimens, I can find the number really dropped to only 12 museums I can find they actually have digitized their bird egg collections. And the number keeps dropping when I examine the digital specimens. Only 9 museums have consistently applied and used a scale in their photos. And the number keeps dropping when I think of it, we consider if they actually has a standard color chart in their digital specimens. And I can only find 2 museums, unfortunately. And that means like only 0.8% of the digital specimens I can actually use for my research. And in terms of the georeferencing, I think most of the museums that have considered to upload their data set to non-local global platform, they also think about to georeferencing their data. So that's about like 17% of the museum. I would also like to point out some other issues as a scientist that are trying to find bird egg collections around the world, because egg collections are often considered as part of bird collections, so they are often mixed with skin specimens, and that make it actually very hard to find them. And I think most of the museums actually, they indicate that's egg collections and they put the information on the column preparations, but that's not a standardized columns. If you look at the content, that means preparation columns means like how they prepare the specimen. So they often content like 10% of alcohols and all different sorts of process or notes they want to talk about. And that make me really hard to find if is this a specimen or not. I think the best practice would be if you could have some kind of code for your category number, for example, EGG. And some museums, they actually have nest and eggs collection together. And so they will use a code such as like EN, which means egg and nest, and that would be fine too. Or some global platform, they actually have some standard column like collection code. So if you could like simply put EGG or EGGS on that code, and every museum could do that, that would be really nice for me to find the collections. All right, so toward the end of my talk, I would like to emphasize again the importance of a standardized protocol for digitization. Especially for bird egg collections, I would like to call for a standard practice for the community when you digitize your bird egg specimens. That means not just the specimens in your photo, but put a catalog number, and also I think a light absorbing background would be really nice to make the eggs stand out, and black recommended. Also always include a scale and the standard color chart, which means not just the color patch, but also the standard gray scale that a factory actually provide reflectance values for those patches. And I'm not trying to give some kind of promotion, but like X-Rite or Calibrite are really recommended. They are really good companies. All right, I know that how hard it is to be a staff in the museums, and there are a lot of challenges ahead. Especially, I know that digitization for the bird egg collections is kind of behind the schedule, but from my point of view, I think it's actually a great opportunity at this point for everybody's to work together and see if we can create a standard digitization protocols for the egg collection so that can be more accessible and applicable for more purposes. And especially now that we're living in AI time, so when we think about digitization protocol, we should think about the whole pipeline from the specimen digitization protocol, and also what kind of computer tools that we can use to extract data from those digital specimens, so we know how to digitize our specimen to make it more useful. And then the last step would be the science research. So if we can think about incorporate all these different steps in our protocol, like when we think about digitization, I think that would be more useful. And to close with the optimistic note, I heard about the DISCO project that UK got a huge grant. I was hoping that even though this photo doesn't use egg photos as their example, I would hope they actually include bird eggs, and I will find some digital eggs from the UK specimens. All right, at the end, I would like to thank the fundings from Academia Sinica and also travel grant from the Spanish Society. Also, all the museums have been helping me along the way, and my lab members from Ecoacoustic and Spatial Ecology Lab. Thank you.
All right, we do have time for questions for Iru. Anybody? Question, go ahead.
Thank you for the talk. I'm thinking about existing egg photos that we have in our collection. If we have metadata in the photos and we know what station they were taken on, what camera and all of that, and if we get a color checker, is it too late to take a picture of the color checker and embed that in the image?
That's a great question, actually. So one of the larger collection is in the WFVZ museums near Los Angeles, and they actually have spent a lot of effort digitizing all their collections. But unfortunately, they do not have a color chart. So actually, I was talking to Renee, who's the curator, and it's like, if we can, put a color chart and sort of use the same settings and take a photo of that color chart and see if we can calibrate. So then we can then use all the photos that have digitized. That's a possibility, but I think we'll probably have to talk to like more photography side and also engineering side or computer staff to make sure that calibration is really gonna work. So we don't sort of sacrifice the value of the science. But yes, I would love to talk to you if you're interested, and I would love to see your egg collections too. And if any of you are thinking about digitizing your egg collections, feel free to talk to me. And I also have everything to go to your museums and digitize your bird eggs.
Okay, you're invited to Colorado. Wait, hang on. Do we have any more questions in Zoom? No? Okay. Also, just one comment. I think all of paleo data folks, we were all thinking of Jacob's talk from yesterday in this session, when you were talking about the problem with putting all the data in the preparations field. We have the same problem for skeletal morphology and for preparations in paleo too. So well, I hate to see the same problem other places because I hate for you to have problems, but it's interesting that you guys have the same problem with the egg data. Yeah. Huh? It's a problemtunity. It means we can work together to solve it. So great talk. Thank you so much. All right. Of course, now okay, well, good. Megan is our next speaker, and she's right here. Sorry, it went away on my phone.
Thank you so much for having me. I'm Megan Lahti, and my colleagues Cecilia and Megan are not here, but we comprise the Natural History Museum team at Truckee Meadows Community College, and we're located in Reno, Nevada. For those of you not familiar, community colleges are offering the first 2 years of what can become a bachelor's level degree. And they often serve as stepping stones for our students. Many times they are non-traditional or are looking for a more economic way to get a higher education. One advantage for students and faculty is that we have really small class sizes, which confers a benefit for education. We have about 1,500 community colleges in the United States. Most of them offer biology programs, but the problem is, excuse me, that so few of them actually have natural history museums, which is odd from our perspective as, museum-going folk. Our research museum is first and foremost a teaching museum, and so students are involved in the process from collection of specimens to curation. And we also maintain a research level collection. And so we want students to understand the prestige of a museum, what that entails, and also learn some of those skills. But we also want to be able to connect with the global scientific community and share our collections with other museums. We've been around for about 4 years, and we've grown very rapidly, which has created a problem, but a very excellent problem if you're in the museum world, more specimens. Students, administration, and community have all played a part in making the museum possible, and I'm going to get to those in a minute. But some of the skills that it's taken to make this happen at a community college have relied heavily on creativity, being a salesperson, persistence, patience, and a lot of caffeine. One of the problems is that if you think of a higher education institution, it's usually a university or four-year college that is naturally embedded with a natural history museum. For a community college, the infrastructure just isn't there or it's not familiar, and so that presents additional challenges, but also additional opportunities. So for students, they are key to helping us develop our collections, but also using the collections. About the time that the museum was forming, we overhauled our biology curriculum. One of the problems we're seeing is that students are graduating from their bachelor's programs with little to no exposure to actual specimens, living or dead. And if you're a biologist or wildlife biologist, that's a huge problem and a concern. So we wanted students to have that exposure and familiarity and to really understand when they say, hey, I want to be a biologist or I want to do biology, that they know what they're saying. And our undergraduate research students essentially act as assistant curators in helping us backlog or catch up on a lot of our backlogged work. We also emphasize at our community college the interdisciplinary value of a museum because for us, a museum is biology, but it goes much beyond that. And so connecting that resource to disciplines other than biology is really important to us. A side step to this is that in creating a new biology curriculum that's more field technique-based, more hands-on learning, more research-based, we developed a wildlife technician skills certificate. And this is getting students in 1 year of prescribed classes to the same level as a bachelor's student, so that when they apply for entry-level technician programs, they're at the same level of competitiveness. From the community, most obviously, we're obtaining specimens, and this is part of our creativity. We went to a taxidermist who gave us 6 large bins of African game pelts that were left over from a World Wildlife Foundation museum installation. We're working with our Audubon Society, private donors. University of Nevada, Reno is down the hill from us, and they're the only university in our region. And so they give us a lot of hand-me-down specimens that will have little to no collection data, but for us, that's great because these are excellent teaching specimens. We learned early on in this process that administrators respond to problems, and so we created a lot of strategic problems that kind of leveraged the museum and its capacity. So we redesigned our curriculum, and that necessitated things like plant presses, ento supplies, field guides. We had student collections that were donated at the end of the semester that require space. So that led to, you're looking at the back of our organismal lab, which is our museum currently. You're looking at cabinets and shelves that are used to store our specimens and our supplies. But we outgrew this before it was installed, which is a really cool problem to have. And so we're getting a small classroom renovated that will be a dedicated museum space for us. Here's where it gets really interesting is with administration in the sense of funding, because we have no budget, but that's actually been a good thing for community college. If you talk to university, no budget means no museum. And so, you know, in 4 years, we've received over $120,000 in funding, which is pretty impressive. I won't go through the areas that this is kind of manifested, but I think it speaks to the power and capacity of a community college to house a natural history museum. And, you know, that it can bring prestige to the institution, the program. Our biology classes are more full, we have more graduates, higher retention, all these measures of success. And they've been really helpful for our entire institution, but most importantly, the students coming through our program. Just a quick timeline. In 2020, as a coping mechanism for COVID, I started collecting plants. And that was our initial run at it. It wasn't until the next year that I started to realize, hey, it's not just botany that should have all these specimens, the other classes should as well. And it just kind of snowballed from there. And it was never intentional to create a museum, but that's what's happened. So basically, you know, no specimen left behind. We went out into the community, tried to get as many specimens as possible. We started overhauling our biology curriculum, getting funding for class supplies. And we also joined Arctos database right away because we wanted to set that nice foundation for research level collections, as well as accessibility to the global community. Unfortunately, at community college, your job is mostly to teach, and that takes about 200% of your time. So the Arctos digitization has always fallen on the backside. And so if you look to the bottom right, this is where it's really cool and shows the potential of community colleges to have these museums, because there's now a coordinator position for the museum because it's tied to the labs as a resource. So instead of being all volunteer time, I'm mostly volunteer, but a little bit of stipend. And this semester, I'm on sabbatical for a project that will be aimed at getting some of our backlog data onto Arctos. This has been a ridiculous endeavor, and I don't encourage it, especially if you're looking for any sort of, I think they call it a work-life balance. I'm not very familiar myself, but you guys are very aware of these benefits that, you know, span from your museums into the community college. They translate to whatever institution you're at. But this has been a tremendous undertaking, and if you look at that timeline and maybe just, you know, draw it out 2 to 3 times as long, that sounds a little bit more reasonable and manageable. One of the things that we really found was in talking to administration, if we presented things from a numeric, a quantitative perspective, that's when we got their buy-in and their understanding, because administrators are more operating at that business level. And we're operating at a qualitative level, talking about how great the specimens are, what they offer our students, you know, the outreach components. So once we started putting into numeric form the benefits of our museum is when we really captured their attention and they started to understand us and started to champion for us. And so we're pretty happy about that. So our website is up and coming, but we emphasize mostly collections from the Great Basin. which is a large vacant space. And we also have a random mix of specimens donated from various entities as well. So thank you very much.
All right, do we have questions for Megan? I love the idea of strategic problem creation. That's amazing. Yeah.
Thank you very much for the talk. It was very inspiring. I just wonder, what exactly do you mean by the research grade specimens, research grade museum, research grade collection?
I don't have any pictures, but it means that we're using museum quality materials to archive specimens, such as herbarium paper, specimen labels that are ethanol proof. We have a professional photography station. We're putting specimens in their correct containers, whether that's, Cornell trays or ethanol solutions. So if you walked into our collection or you saw some of our specimens, it would be similar materials to what you would find at a museum such as, Smithsonian or University of Nevada, Reno, for instance.
I think we have one question from our virtual audience. Sorry, Deb.
This question came up earlier, but I think it actually is a good one to ask you too. On Zoom, Esteban asked, how do you prioritize which event meetings to go to? And I think that's an important question for you because I can imagine you have many polls. And so thank you for coming to Spinach Tadwig.
Yeah. Thank you so much for having me again. It was Japan. No. I understand this is being recorded, so if you could please remove that. No, we have had a really great relationship with Arctos, and so we're trying to put more buy-in and effort into this community because if we're trying to maintain our museum at research grade, that's where I think we have the most knowledge to learn and gain. And so this is the place to connect with you guys. It's been a great conduit to obtain specimens that you guys are trying to offload, but also connect with the museum side of what we're doing.
Hi, that was great.
Thanks.
So in the interest of being strategic for this community, broadly speaking,
Yes.
did you know of other community colleges who've done this? Did you have any contact or, allies?
Allies, yes, but no contact or context. This came out of more than anything. I've worked in museums as volunteer, but also as working in different collections. And so that has been a personal interest and valuation on my end that I kind of imparted and saw as a natural convergence with our institution.
So I think it's a great subject for a paper.
Oh, yeah.
Yeah?
Yeah.
And I think at the same time, I can think of at least one other place that I know of that would have really compelling model to share about how they had success doing what you're talking about.
Okay,
Becoming relevant to an institution in ways that we may not have been doing in the past. So I think that would make a very compelling publication and collaboration. If you're interested, I
Yeah,
can point you.
absolutely. Thank you.
You're welcome.
Yeah.
Any other questions for Megan? Yeah. And we can come back to this too at the end of discussion, but Ryan has some community college.
Yeah. So this is less a question and more of a comment, but
Okay.
one of the best things that you can do in a small collection is just put your name out. So join an aggregator, make sure you're registered on Index Herbariorum. Even if you're not sharing a ton of data, share it somewhere. Just make it so that you're available, because what happens is when people are trying to put together projects, if they can't find you, they can't include you.
Yeah, absolutely. Thank you.
All right, I thought Ryan was going to talk about community colleges, but I was wrong. I was wrong. Okay, thanks, Megan.
Thank you.
Okay, I'm just going to make one more comment since I have the mic. I love the idea too about incorporating the digitization from the beginning as students. Like, I think that's so important for the theme of this session, and bridging the gaps is just making that part of the whole data life cycle. So I love that. All right, our next speaker is Jen Zaspel.
Thank you very much. And, ooh, 1 minute and then I can say good afternoon. According to, oh, yep, here it is. Good afternoon, everyone. As Talia said, I'm Jen Zaspel. I'm from the Milwaukee Public Museum. I'm one of the co-lead PIs on the terrestrial parasite tracker, TCN, along with Dr. Stephen Cameron from Purdue University. And many, many others, which is why I've just said plus TPT network. We're quite a big group at this point. So I'm here today to share a little bit about TPT for those that may not be familiar with the project. I'm also going to give some highlights as we're kind of winding down in our work. And with that, I wanted to also share some of the resources that the network has developed within the context of sustainability that we all hope will help facilitate discovery, learning, community engagement, and even future digitization projects for not only this community, but beyond. There we go. Okay. So, terrestrial parasite tracker, TPT, we are digitizing collections to trace parasite host associations and predict the spread of vector-borne disease. This project started in 2019, and we've just been approved for another no-cost extension. Several of our collections have completed the work. There are still some that are just still getting caught up from the disruption of COVID right as this project began. And so we do hope to be finished next summer. And so when we got this award, it was part of the ADBC program at the National Science Foundation, which is now Infrastructure Capacity for Biological Research. And here on the left, you can see a picture of the less charismatic insect groups that were the focus for this work. And the number of species within each of these various orders is sort of reflective of their distribution in North America, north of Mexico, which was kind of our geographic scope for the project. So we had 24 primary data providers, including 2 pens, 11 collaborating institutions, 7 supporting organizations, so organizations that participated in the project and contributed data or other resources, but maybe were not, you didn't have funding from NSF. We had 36 co-PIs and leads on subs. And some of you may have heard me say this before, but I'll say it again. We were so proud to have 13 PIs on this project that were new to the ADBC program. We had mammalogists, I'll say half. We have mammalogists, entomologists, parasitologists, biodiversity informaticians, folks working on endoparasites, ectoparasites, folks at small, medium, and large universities, folks at small, medium, and large museums, and folks that were, early, mid, and later career. And it was just such a wonderful group of people. And I guess I keep saying was because this is the last time we were all together in person, which was in 2019, just before COVID happened, at least together in person. So now we're up to well over 150 participants in this project. It's really just too many to count at this point, but again, just a wonderful group of people and a really successful project. In terms of justification and scope, as we all know, these organisms have the potential to inflict an enormous burden on vertebrate health directly and as vectors of human and animal disease. There's also some substantial organismal diversity within what I guess I'll call this ecological niche of organisms. And parasite groups have historically been underrepresented in terms of the digital or digitized collection space. And so when we went to scrape data online to see, what the primary aggregators for entomology and even beyond had for these different groups of insects, it was really, really kind of shocking. And so you can see where these heat maps are showing, collections that had digitized their specimens. They're lighting up red hot, but if you look at those number of occurrences, it's actually, not very many specimens. And so with that, and also the great potential for this data to greatly enhance downstream research and education and discovery. These were the merits that we assigned when we were putting, or that we focused on when we were putting the proposal together. We put a heavy focus on biological associations and really having a better understanding of parasite-host associations and interactions. We also felt that this data could advance the field of disease ecology through the creation of digital records for organisms that spread disease, and just kind of allowing us to better understand the ecological interactions with having better georeference data sets. And of course, we also wanted to use those georeference data points to create distribution maps and hopefully help the various disciplines identify areas that might be threatened by parasite diversity, or the reverse, or maybe related species in need of conservation. And of course, the data, we hope facilitates, comprehensive systematic approaches and alpha taxonomic studies through the images and other resources that the project developed. So these were our objectives, which were to digitize 1.2 million parasite records from across North America. We had a plan to share the data with aggregators such as IDIG-Bio, SCAN, and GBIF. And we also set out to capture 500,000 new parasite host association records in GBIF. And on the right, you can see kind of a high-level TPT digitization workflow. This project was focused on digitization of specimens in ethanol, specimens on pins, and specimens mounted on slides. So kind of an anything and everything goes approach, not only in terms of the material that we were digitizing, but also the various relational databases that our providers chose to use for data transcription and other information. And then from there, as I mentioned, these were the main data aggregators for the project. Some of the collections that were being hosted by SCAN have now moved to ICDISIS, so I should add a little ICDISIS logo onto this workflow. And in terms of highlights, as of now, these numbers are going to be a little bit out of date just because we're right in the middle of our project reporting period, and so we're just gathering all of the new data as we speak. But as of last reporting cycle, we had over 800,000 transcriptions and images. And these are some heat maps that our project manager, Erica Tucker, put together, just sort of showing how far we've come since we submitted this proposal. And this is the heat map that I showed earlier on in my presentation. And then this is what it looks like now. You can see that we've got a lot of additional records. This is for ticks that are not only from TPT organizations or participants, but we've also, I think, gained a little bit of momentum even outside of this TCN network in really getting folks that maybe are not part of TPT to also get their parasite records out there. So we have exceeded our goal in terms of association records that we wanted to have in GBIF, and we've also reached almost 170,000 notes from nature data transcription, so 40 expeditions, many of which have really fun titles, like Jumping into the Field Museum Flea Collection, Mite at the Museum. Another one that we relatively recently completed from MPM was called a Movable Fleece. So yes, very creative and fun group. And 3 of our collections not only reached but surpassed their digitization goals. So congratulations to folks here. And there may be more to add to this list once we get through our reporting period in a couple weeks. All right, so now I want to talk a little bit about some of the resources that the network has helped develop that sort of fall outside of the digitization goals that I mentioned in the previous slide. And I'll go through these quickly. One of the big things that we'd decided to focus on when folks kind of had access to their collections disrupted during COVID was taxonomy. And so we've put together a lot of taxonomy resources that you can access through our GitHub site. Our student, Kat Sullivan, who's here, had a poster talking about some of the taxonomy work that was produced out of TPT, and she's also published a paper on that this year. So, but basically this work has resulted in taxonomic names lists, with expert contact information, and we are still working, to some extent, to encourage folks to keep these resources as up to date as possible. But you can nonetheless, check in with the expert that's listed to confirm if you are interested in any of the nomenclature for these groups. We've also used a lot of the knowledge that that we've gained over the course of this project in terms of species interactions and association data to put together some training materials. And we've used these training materials in the entomology collections management workshop. We just hosted another one just a couple weeks ago at Yale Peabody. And we have this workshop on host association data, not only available in person during the collections management workshops, but there's also an online tutorial that could be used in other workshops and for other groups, and even in contexts outside of entomology. We've also put together a tool to track digitization progress. This was a script written by one of our curators at the Milwaukee Public Museum, Dr. Nick Dowdy. And basically, this is a tool that allows you to put in, what your digitization goals are for a project, and then sort of allows you to project where you might expect to be given the sort of task analysis or pace at which you are digitizing your collections. And this example is one using scan slides. We've also shared a lot of resources in terms of workflows and imaging, georeferencing with the bug flow group. And so this is another group of entomologists, but they've done a lot of great consolidation of digitization work over the last several years. And so a lot of the TPT resources can be found on their site. And sorry, I keep pushing the wrong button. So finally, this is just a list of some of the different things that, some of the different resources that TPT has developed. This is not a comprehensive list by any means, and we did have a QR code, but someone mentioned to me that it wasn't working. So if you're interested in any of these resources, whether it be for digitization, teaching, research, or community science projects, please reach out to myself or Kat, or there are others here, Craig Brabant and Dan Young from UW-Madison, that are also have been a big part of this project. And I think I'm out of time, but thank you to all of our partners and funding, and thanks for listening.
All right, that was great. And if you have questions for Jen, we can ask them in the discussion. I don't know if we have anything on Zoom, nothing on Zoom. Okay, great. Our next speaker is Peggy Newman.
Hello, everyone. Taking a little bit of a turn off to the side and going over to an aggregator rather than a collection at the moment. I thought, given the conference today, I'd talk spinach and tadwig. conference, I'd talk a little bit about services that the ALA offers collections. So I'm Peggy Newman, I'm the data manager at the Atlas of Living Australia, and I don't think we've got it here, but I thought I'd mention, I haven't given a lot of background on what the Atlas is, but we're hosted at CSIRO in Australia. We're a research-funded organisation, and we're the Australian node of GBIF. So before I start, I want to acknowledge the traditional owners of the country that I live and work on, the Wurundjeri people. I pay my respects to their past and present elders, honour and celebrate the profound and continuing connection of traditional owners to land, sea, sky, waterways and biodiversity. And the Wurundjeri people have this phrase, Wimajeka, which means we meet with purpose. So today I want to cover a little bit of background on specimen data in the ALA and services that we have for collections, so standard search and download stuff. And I'm going to focus a little bit on our user annotation system, by which ALA users can give feedback to dataset managers, and also a bit about measuring impact and the kind of stats that collections can use. And also just some next steps, what we are thinking about going on to next. So data in the ALA comes from a variety of sources. I think I categorise them as about 4 major different sources. So we've got our citizen science colleagues who give voluminous and reasonably up-to-date data, the eBirds and BirdLife, iNaturalist, etc. The ALA has relationships with all of Australian state jurisdictions, so New South Wales and Victoria, who, for example, deliver their environmental assessment data to be openly shared. We have our research community, and there we've got OBIS, who are we've got the good fortune to be co-located with in the CSIRO. So our OBIS Australia node and us work closely together to mobilise Australian marine data. And finally, the stars of the day, the herbarium collections data. So the Australasian Virtual Herbarium was really preceded the ALA and the online zoological collections. Make up around about 12 million of our specimen records, and they indeed were the original business case for the Atlas 12 years ago or so, was based on delivering online access to those collection records. But we've got about 15 million specimen records in the ALA, and you can see the global reach that those institutions have. But the other thing that's reflected in that numerical difference is my team has been working more recently with a lot of smaller university collections and small government collections as well. So that's been really encouraging to seeing them come on board and share data. We have search tools, ageing a little bit at the moment, but nonetheless you can search on collections in the ALA through taxonomically, through location or just text searches. We've got around about 190 collections. And I just wanted to draw your attention to what a preserved specimen record looks like in the ALA. So we've got some dataset metadata there. There's a lot of similarities here to GR-Cycle, and a little bit more on that later, but you can see here on the 3rd and 4th row there, we've got our institution code and collection codes. So our museum, Queensland Museum is entomology collection here, so if they provide in their occurrence records their institution code, then we can map that to the actual institution record that we have in our metadata registry, and then link across to the metadata pages for that institution, and likewise with the collection. So we're tracking the data for each collection in that manner, and that link will link across to this metadata page. As I said, the metadata page contains the descriptive information, the contact information, and a link out to all of the records. And that metadata is curated by each institution. So I want to move on to talk about our user annotation system, which helps get feedback from users back to data owners. So we'll walk through this workflow where a user can annotate a record, the data provider is notified, they can take action, and then somehow resolve that problem. So first of all, what it's called on the front end, in that screenshot above, you can see each occurrence record in the ALA has a flag, an issue button. And we can click on that and be invited to categorise the flag, the comment that we're going to make. So it could be a geospatial issue or a taxonomic issue, etc., etc. And add a comment and hit submit. And then that flag will travel along with the record. So we can see here in the example, this user has come along back in 2020 and they've said, okay, so this species, the text locality says that this is in this place, but the coordinates and the map are incorrect. This record needs to be fixed. So a couple of things happen then. An email sent off to the data provider, informing them of the problem, but we've also got this data quality profile that we use to filter out records, and what we do is we hide the record from the default view for the ALA user. It doesn't mean that it's disappeared, it doesn't mean that it's deleted, it just means that to get to the record you have to actually click through down here. You have to click through to say, yeah, I want to see the records that have unresolved user annotations. But it means if there's a bodgy record that has got a funny location on the map, it comes out of the map until it's resolved. So then the data provider has some options. They can sit for a while in this default status where it's an unresolved issue, and I can see that it's an unresolved issue. They have the opportunity to disagree. I've looked at this record, actually it's correct, so I'm not going to do anything. Or they can go and correct it with a data refresh. So we've been working for some time in our data team to make it easier for collections or for any data providers to fix their data at source and provide the data again. And that's what's happened here. So on this record now we can see the data providers come back, they've said, yes, we've fixed this in our EMU system and we've sent the data through and now it's resolved. So once they've done that, the issue travels along with the record. We can see that little workflow, that comes with the record. We see that happens, it gets included back in the default view and everyone's happy. So that's I think a lovely system that we've had for a long time. It's business critical for a lot of our collections, and citizen science agencies have also been interested in using it, and I think it's got a lot of potential. In terms of impact, measuring impact, so we've got, I think, two really useful ways that collections and institutions can use ALA information and information from ALA and GBIF to measure the impact, to demonstrate impact of their collections. So ALA collects usage stats. Now, having those collection institution codes means that we can collect those stats for those particular collections. So what we've got on this metadata page that I threw up before, we've got a usage stats tab, and we have these download statistics. A couple of things. The download statistics are categorised. We ask, when somebody does a download, we ask them for a reason that they're downloading. And one of the nice things about these reasons is that we haven't actually changed them for around 10 years. So, so you've got this really nice solid time series data that has started from the moment the collection came into being and occurrence records went in. That says the last 12 months and all downloads. Basically, down there in on the screen, you've got download usage stats, and that downloads everything per month. Cool. So, so that's some nice, solid, reliable statistics that can be reported and relied on. that your bean counters should appreciate. But likewise, that's at institution level as well. Like I said, the collection code enables the collection stats. This enables the institution stats. So that's independent of the actual data set entity that came in, the data resource that was loaded. Cool. And then the next way to measure impact is using GBIF citation tracking, which is another story again. So GBIF also track download counts, so that may be another thing, but yeah, again, some nice time series data that can show how your collection data is being used. For us now, I think it's become fairly clear from this conference that we need to align this stuff with GR cycle. So some of the collection codes are older and don't line up, and we want to work with our collections to tidy that up a bit so that that information all travels through both those systems really nicely. Not a difficult technical exercise, just a people one. And we're working too on repatriating some collections data to Australia from international collections. So we've got a nice little bit of software that we've written that'll go to GBIF and package up stuff from other natural history museums and bring that back into the ALA. And also the ever-present problem of specimen linkages is something our local collections community is screaming out for. Thank you. That's all I had for today. Thanks for letting me come along and talk. Any questions?
We have time for questions. Marie, anything? GR cycle? We'll talk. Okay. Anybody working on MIDS? I love the idea of this like data quality profile, and like I'm thinking about how you could link the data flags and like a MIDS profile. This came up at a paleo meeting we had earlier in June. There was a talk about research quality of fossil data, and I love the idea of looking at that and having, well, here are the flags and here's how complete the record is, and maybe it's appropriate for answering, I need a very complete record to answer these type of questions, or whatever. But I love that idea. Yeah, Deb.
Yeah, data quality for us is a question of fitness for purpose, and you can't. So what we wanted to do was try to hide some of the more problematic records and make it so that people had to work or click through and say, yes, I want to see the problem records. I want to see the spatially problematic records. I want to see the taxonomically problematic records.
I love that. I mean, this notion that our data is in rows and columns, and I'm always looking for more ways to visualize those things rather than having to sort and try to filter, but you're providing a doorway if people want to do that. So I know in TaxonWorks we're trying to move forward, and we've made some steps toward, not MIDS yet, but this notion of what the GBIF asserts about your data, right? So some people in here have heard that already, but we get that data for you now and bring it into the database. We have soft validation already that supports nomenclature to help you not go wrong when it comes to nomenclatural data. What we want to add is this so-called soft validation, so that when GBIF does assert your coordinates are flipped, or this taxon name, we've never heard of it before and we can't index it or whatever, that you would then be able to do the same kind of thing, either show me all records where GBIF asserts this is a problem, that kind of thing. So we've got the feedback already from GBIF built in, so you don't have to go out to get it. So the next thing is to be able to act on it on the individual record, or group the records by what GBIF asserts. They've already done the hard work to figure out an algorithm that finds these things, so we ought to make it possible for the people who are in the databases to do work there. We also have a completeness strategy that we started a long time ago that gives you some idea against MIDS. But that's another question at the end about skeletal records. How do you know which ones are skeletal that you took, and how do you find them?
All right.
Cool, thank you.
Wait, I have one really quick question.
Okay.
Is there an atlas of fossil Australia, or an atlas of dead Australia? Because we talked about a catalog of death at the catalog of life meeting for fossil taxa. And sorry, this was just an aside, before you step off the podium.
There was, and there's not anymore.
All right, and our final speaker before discussion is Chris Hunter.
Hi, my name's Chris Hunter. So I'm slightly unusual here that I'm nothing to do with museums, libraries, collections, or indeed biodiversity, because I'm a publisher. So my background and the reason that I've sort of involved with this is partly because I'm involved with the Genomic Standards Consortium, so I'm a geneticist by training. And the Genomic Standards Consortium obviously produces standards to do with genetics, but they're also overlapping because they have sample metadata included with them. So we've worked with the likes of TDWG to try and produce a sample, some sort of mapping between the two. So I've sort of come into this arena, if you like, via that route. But my day job, as it were, is actually in publishing. So I'm the director of GigaDB, which is a database where we host all the information associated to our scientific papers that we publish. So GigaScience Press. Okay, let me start with the slides. That'll be useful. GBIF, I'm assuming everybody knows what GBIF is, so you can read it if you like, but that's it. GigaScience Press, however, I'm guessing most of the audience probably don't know what it is, so I'm going to tell you a bit about it. As the name suggests, it's a publisher. We've got two journals, GigaScience, the original one which was launched over 12 years ago, and a new one called GigaByte, which was launched about 3 years ago. And the sort of unique selling point, if you like, about our journals is the fact that we have this database called GigaDB, which is what I look after, where if the data that is published or mentioned or used in those papers is not available in a public repository elsewhere, then we will host it in GigaDB and index it and put all of the metadata on it. And we've got a team of curators that will make sure that all that information is appropriate and correct. So what did we have to do with GBIF? Essentially, there was this challenge out there that vectors, or the less charismatic, I think Jen called them, creatures, are not as well indexed, if you like, in a lot of these resources. So WHO and other health organizations are keen to make sure that people can track where these vectors are, where they're going, what the evolution of these things are, if you like, and where they're going. So we want to try and help mobilize the data. So one way of doing that is to incentivize. The incentives in the academic arena are publications. So that's where we come in. GigaScience Press offer the link, so we can offer a paper, if you like, describing how you've digitized these things, how you've shared this information. So you get a data paper, and it is directly linked to, in this case, GBIF for the actual data. So we don't host the data in GigaDB. However, our curators did manual checks and audits of the data that was going into GBIF to make sure that it actually did match. So these things that GBIF flags as errors, we made sure that the authors were aware of them and fixed them where they could. And that's essentially where the collaboration went. So a little bit of background, I think this slide has already been shown elsewhere, but essentially the malaria prevalence is that big red patch across the middle of the globe there, and then general biodiversity recordings or records that are in things like iNaturalist and GBIF and all those sorts of things tend to be in the more westernized areas, not across the middle where the malaria is. So, okay, this is not specifically for vectors, but it gives you the idea that we need to increase the number of records that are in the right places. GBIF introduction, I'm hoping I don't need to do that. Essentially, they've got records all over the world. So how do you increase the biodiversity data in the relevant places? Well, a lot of the times it's things like iNaturalist and eBird, where the people, the citizens will go out and find these things for you. The less charismatic creatures, such as the ticks and fleas and mosquitoes, tend not to be the ones that get into iNaturalist as much. So you want to encourage other ways of getting that out there. So citizen sciences are perhaps, well, I'll show you in a minute. There are some that are doing it, but it's not as common as others. So a lot of collections already have lots of insects, including things like bugs and ticks. How do you mobilize those? Is there an incentive to get audience to do it? So clearly there's already people doing it, so we want to try and incentivize that as much as we can as well. And publications is, for better or worse, the academic monetary system, if you like. So essentially there was a collaboration, or a call put out for a collaboration between GBIF and journals. So GigaByte was the one that was selected. And it was funded through the WHO, through the Tropical Disease Research Organization. And essentially what that means is there was a small amount of money given that meant that the APCs were all covered. So the researchers, the institutes didn't have to pay to actually publish this stuff. It was just paid for by the TDR. And if you want to find more about it, the series number there, so you just go to the Gigabyte website and just find the thematic series and all of the papers that are associated with this series are available on our website. So what was the scope and the coverage and what would we want? So the data had to have a clear relevance to vectors of human disease. So it wasn't just any old paper that was digitizing something. It was about human-borne vector-borne diseases. We wanted to encourage large-scale data sets because one of the features of GigaScience is, well, when we started 13 years ago, Giga was big. So yeah, we were interested in big science. So that was sort of our remit. We want big data sets. So we set an arbitrary limit of 5,000. We didn't stick to that rigidly. It was just, there are some that are smaller than that. But essentially, it was just sort of encouraging big data sets to come in. And it was run from 22 to 23, so it's finished. Well, the funding for it is finished, but the thematic series is open. So if people have got data sets that they want to publish through us and data paper, then that's available. The APCs are very low anyway for Gigabyte, but if it is a problem, talk to us. We can find ways to assist with that. The other point that we made is that GigaScience is all about open data. I don't know if there was a few of these nice bottle openers which has open everything on it, because GigaScience is all about open everything. So there was about 20 of these out there. I've got about 3 left if people are interested and want a bottle opener. Where was I? Yes, it had to be under CC0 license or public domain waivers. So basically all of the data is out there and it's all openly accessible. And one of the things that we did as part of this, the GBIF actually ran a specific support portal for this series. I think they've closed that portal now, but the GigaDB team, myself and my team are able to help and willing to help because that's part of our job. We assist authors in getting the data out there to make sure that it is reusable in every way possible. Fair, for want of a better word. It was very successful in the first round, so it was extended into 2023. So again, there's a write-up about it there on the blog on the GBIF website, so if you wanted to read about the extension. And then about what we actually found. So the dots on this map are new records that were put into GBIF via these publications. So there's a series of different sort of vectors there, but essentially you can see that it's actually covering the regions that we were hoping it would cover, as well as a few additional extra ones in Europe and the US. So some numbers and pretty pictures. It was across 70 different countries, more than 670,000 occurrence records were added to the databases. Everything is open domain. And interestingly, it came from the bottom there, it shows you there was imaging data, there was DNA barcoding data, it came from citizen science projects. So we got a good broad scope of different ways that these data could be, the data could be coming from. So I think it was a good success in that way. Just a couple of examples. So this one is, it's Gigabyte 54 is just the number that's given to the manuscript, but you can look it up under the DOI at the bottom of the page. And essentially this was a citizen science project where they were looking for, there's a tool out there which mosquito alert. I think if you look for the app on Google Apps or the iPhone equivalent, it's still available and it is functional worldwide. So essentially citizens can report mosquito occurrences. So if you get a mosquito bite and you manage to slap it, you can take a photograph of the dead insect and say where you were, when you were, and put it onto there, and you can share that information. So you can actually get real time, well, not real time, but you can get information put out there. So this was one group that's doing it. So it was one of the ones that was sort of Europe-based. So I think the app was originally started by a group in Spain. So a lot of the uptake was in Spain and then Europe. But it's available worldwide, and you can download it and still use it. So that's a little advert there. You can go and do that. And then some of the data was from historical records. So in this particular instance, there was a researcher that kept extensive notes, and they've digitized these notes, and somebody has actually transcribed all of the information from those notes to tables of when these mosquitoes were found. So there's a whole bunch of mosquito occurrence data that's there, which is provenance by the fact that this person has identified them. I don't think there's pictures as such, but there's records to say that they're there. So again, there's more information that's been put out there on historical nature to see where these species were and when they were. We wrote a blog, well, we wrote a paper on the lessons learned from this. So if you want to know more about what we found out, and essentially the summary was that there's a lot of researchers out there that didn't realize you could publish a data note on these things. So you can actually get the scientific credit just for putting your data into these databases. So that's one of the lessons learned. And also it was, there was a lot of positive feedback from the authors, the fact that myself and my team and the GBIF team were there on hand to help with something that is not necessarily a thing that people would normally do with their data collection. So there's assistance out there. Just come and find us, and if you want to talk to me about it, then I'm here. And essentially that's all I have to say, but thank you for listening. Thank you for the organizers for bringing me here, and any questions?
Any questions for Chris? Anybody want one of his coveted 3 last bottle openers?
There's also Pandalorian stickers if everybody wants a little sticker of our...
I already got a Mandalorian sticker.
So
All right, we have time for discussion, or Deb, come on up.
So as you experienced this data, I'd be curious to have some more time to talk about, did you see gaps even in the current data that we could be potentially doing differently or collecting differently, or, you know, data that you wish you had, a column that doesn't exist, with if we'd known this, we could have used this data. I realize for legacy data, that's not so much where I'm going. I'm talking going forward.
So I'm not really the right person to ask. I mean, I look at the data that's there,
Sure,
and fill in the gaps that are there, but I'm not familiar enough with the research and the environment to know what's missing. So,
fair enough.
Yeah, I can't help on that one, I'm afraid.
Oh, that's right. That's right. I asked because I'm also on a grant with another PI where we're looking at the idea of prevention, mitigation. How can we, in terms of the next pathogen endemic or pandemic sort of idea, how can collections be relevant to that and be ready for such a thing? And so we're asking those kinds of questions. So thank you. Right.
Thank you.
All right, so we have time for discussion before lunch. If anybody has questions, comments, thoughts. Yeah, Ryan.
I figured I would just follow up on what Talia was alluding to. So I was the manager of the Southern Rocky Mountain TCN projects, the thematic collection networks. And when we were setting up our project, we actively sought out trying to locate every single herbarium that was in the Southern Rocky Mountain region. And one of the things that we found as we started making our way through and digitizing some of these small, maybe even tiny collections, is they were hyper-focused and hyper-unique. So in a lot of instances, some of these community college collections or teaching institutions maybe only had 4,000 or 5,000 herbarium sheets. But then when we started to look and see, are these collectors represented at other locations? What is the effect of these specimens on just general distribution? In a lot of cases, they might have been providing one of the largest data sets from the individual counties. And in a lot of cases, the collectors that were found in these institutions weren't represented at other institutions. So I just want to get a shout out for the smaller collections. When you're thinking about these and when you're trying to include collections in the project, there's a tendency for us to kind of focus on all of these big institutions because you can bring a lot of data in quickly by just including a few people. But really, there's a lot of information that's being kind of lost or missed by not including community colleges and teaching colleges. And I would strongly suggest that you try to focus on that as you're working through. So,
Thanks, Ryan. That's a really great point. And I think Samantha and and Jen's talk from yesterday, or Katie's talk, sorry, thinking symbiota and herbaria. It's been a long week. The symbiota talks from yesterday really hit on that as well, I think, and bringing lots of collections in. And I think that's one of the things I've been thinking about a lot is how do we make sure that we're bringing everybody along and we're not leaving behind small collections because they do have really important data sets? Or how do we make sure we're not leaving behind even collections at medium-sized institutions? So other comments, questions? Yeah, go for it.
Yeah, I was reminded of a program, and this is leveraging off of what was just said, and also the comments earlier about getting collection managers able to, or collections people, able to visit other collections. The limitation, of course, is you need a little money to do that, no kidding, but not much, let's be honest. If there were a fund that Spinach or another organization could host, that collections people could apply for very small grants to visit other collections. I think that could be a really, really fertile way of doing that, because it also raises the consciousness that that could be a useful thing, and that is valuable as well. But I also wanted to comment on, as we were talking about this issue of trying to incorporate smaller collections, there's the awareness and capacity issues, of course, that go with that. A professor in a community college may not really be embedded in the collections community, strangely enough, because they do have a day and night job. A kind of program that might be an analog to a program that we did at the Natural History Museum having to do with the Dig-In digitization project might be an interesting way of doing it. What we did is allied collections staff with nearby educational institutions and brought those pairs of people together for workshops together in groups. We brought them to Los Angeles because that was convenient. And that is, in a way, creating a localized community connection between museums and, in this case, educators, but could be other organizations, but also creating a nationwide network of such people because they all got to meet together for a summer. And so some kind of program like that might be an interesting way of trying to address the issues that you're talking about of bringing these people together.
Great. Thanks. Hey, can you say who you are for the folks on Slack too, or on Zoom? Sorry.
This is Dean Penchef, Natural History Museum, LA County.
Okay, thank you.
Sorry.
No, you're fine. Okay. Chris, go for it.
Hi, sorry, I just want to follow up on that. So I don't know how many people in the room know about biocuration. So it's a society, the ISB, biocuration.org is a website, but they actually do host a fund for travel grants, micro grants. So if people want to go and do biocuration somewhere, there is at least a possibility of getting some money from the Biocuration Society to go to other institutes. I think it's only like 1,500 euros or that sort of price range, but that's probably enough to get transport. Yeah. But anyway, I just thought I'd plug that for you.
That's great. I didn't know that existed. Marie, yeah. No, go.
So actually I have a somewhat
Wait, can you say who you are and where you're from?
Hello. My name is Marie Grosjean. I work at the GBIF Secretariat and I work with the Global Registry of Scientific Collections, which is why I was going to ask. So I get that it's maybe when you're in your local area, you kind of have an overview of the small collections, right? But at the global scale, like I don't know small collections myself, and if they're not on the GR Cycle or any like online website, then I cannot guess like names and localities and everything, right? And so my question is like, could there be a way to like make them more visible? Maybe, I mean, of course, I want the information in GR Cycle and the Global Registry of Scientific Collections to be up to date and everything. And my preferred way would be if people could just register their collections there. But even if that weren't the case, like, how can you like make those small, local, maybe personal collections like just like a bit higher so that we can see them? And I think in the US, a lot of people are very good at sort of be online and have strong connections with their own communities, but it's not the case everywhere in the world. And it's very difficult in some places to even have like bigger collections like being visible. So if anyone has any thoughts on how to do that and how to improve, I think that would be great. Thank you.
Great. I just had a thought. We should have like some kind of, you know how we have a bio blitz? We should have like a collections blitz at every meeting we go to. and get people registered. Like when you go to your society meetings, like maybe we could do that.
Yes.
Okay. I'll send you a Slack message. Okay.
I think that's also a bigger topic for when we talk about visibility, our need for marketing and communications for when Marie says, how do we do that? How many marketing and communications people are in the room here who can tell us how to amplify our own voices? Yeah, none, right? So this is a challenge. For me, I would say perhaps, I'm not sure of this perception, but in my talk, I was at a discipline-specific meeting. I'm trying very hard not to say the discipline.
You can say it.
Really?
Yeah, go for it. Why not?
Okay. There you go.
This is a safe space.
All right. Well, I hope so. Here we go, folks.
I mean,
So
it's
I'm at the American Society for Mammalogists meeting. So here's the story. I'm going to the committee meetings, and some of you who know me, this is going to mean different things for those of you who don't. So there's an informatics committee. Oh, I'm going to go to that, right? That's me. That's my thing. And another colleague from the University of Michigan said to me, "Oh no, Deb, no, don't go there. They think that informatics is building a website. They have no clue about TDWG. They don't know about standards. They don't think about data sharing. They're not thinking about FAIR. They have no clue what the bigger context of that word is all about." They also have built, anyway, it turned out to go to the systematics committee meeting is where I went. So then the next story is, in short, for years they have been publishing where are the mammal collections of the Northern Hemisphere list. How do they publish? On paper. This is the first year, 2024, that they have decided they're not going to publish on paper. So then I'm sitting in this committee meeting, and the next thing that came up is, we're going to build on our website a list of all our collections and where they are. I'm like, okay. However, kind of GBIF's already kind of got it, and they've got an infrastructure, and there's issue tracker and pull requests and all this cool features. And I basically told the story in the nutshell. I said, do what you want with your money, your hard-earned money for your society, but at least talk to GBIF first. I gave them your name and the other friend, Kat Chapman's name, and said, look, they have an API, you have an API. If you're going to build your own, at least talk to them first so you can make sure they're going to work together and you've got the endpoints that you need to share information. But why was I at that meeting? It was completely happenstance for a completely different reason, a completely different workshop that we were running about pathogen data and collections. So there was no other person in the room who would have brought that conversation to the table. And so, are all these small collections getting their visibility? Do they know what their power is, even in the US, to have the data we get back from being inside GR-SciCall? And that was one of my points in my talk was we need ways to be strategic. And that means going to the leadership of these societies and realizing that leadership needs to understand they have these gaps, and they don't. And so what parts can Spinach and TDWG play in that kind of, so we're not just going and having an instance of a workshop or an instance of a help desk. It's how can we put a strategic effort into making a cultural change.
Yeah. Deb, okay, we're going to keep going because just before you respond, somebody else had a question. Yeah, I want to make sure you get your question asked. And could you say who you are and where you're from?
My name is Alicia. I'm from Seattle, Washington. And I from a company in Seattle, Pacific Technology and Resources. I actually did a presentation of our application that our company developed. So just to answer Marie's question regarding, for smaller, say, herbarium collections, how to make them visible. So this is the app that our company developed. It's called eHerbarium. I think this is a great tools for a smaller collection. So say private collection, they can take their, the app has the ability for you to take photos of your herbarium collection and give a taxonomy details and putting notes in there. So I think, the tool is free. So it will be a great way for those smaller herbarium to have visibility to the public.
Okay, great. Thank you so much.
That's all.
Okay, Holly and then, or Jen, do you want to, Holly had something from the chat first. Oh, it's from Holly, sorry. And then Jen.
Holly has something from me. No, I was just gonna tie this kind of back to where we started in the session yesterday, a little selfishly. I think part of what we're also trying to do to help with this change, we need strategy, we need very specific resources and capacity and people in strategic positions to move messages forward, culture change, all that. However, I also think part of what we're trying to do is building this shared knowledge network, right? So even though that was happenstance, we want those networks to be there so that when the time comes for someone to be able to share, oh, have you heard about GR Cycle? Then they're there and able to do that. So I think being able to do it in both ways is really nice. And that's part of this kind of shared knowledge ecosystem that we're all looking at building. And it was really nice to see the very many different versions of a knowledge model, well, I can't talk anymore, shared knowledge model in these other networks that everyone has presented on.
Yeah.
Thank you.
I agree with Holly. Okay, Jen.
So just really quickly, Jen Zaspel here at Milwaukee Public Museum. I wanted to share another set of allies or people who can provide resources and support in many, many different ways, and that is the Natural Sciences Collections Alliance. And so please check out their website or come and talk to me. A lovely group of people that are on standby waiting to help, all collections and collections worker. A Natural Sciences Collections Alliance, often called the NSCA.
Natska, yeah. I think Lindsay Walker, who is our fourth session organizer, who is not here, but she's on Zoom. I think she's putting a lot of these resources in the Oxford Abstracts chat, and I know she put a lot of links in yesterday. So if you want to go back and find anything from our session, I'm just being mindful of time because my tummy is grumbling and I'm sure everybody else is hungry too. I'm a collections person and I get in early and eat at like 11:30. So this is a very late lunch for me. Did you have one more thing to say?
If you know of additional knowledge networks or models for how to do distributed shared knowledge, please let us know because this is what we are trying to find and document and understand. And now Jesse has an announcement.
This is just any registrars or collection managers who hold registrar hats or anyone interested in acquires, accessions, loans, disposals. We're going to do a lunch, gather near outside of this door and grab a lunch and go find a spot to chat and talk about all the things, but also get anyone. So if anyone's interested, wants to join, please just come find me or drag someone over to us. Thank you.
Great. And I just want to thank all of our speakers from yesterday and today. This has been great. And let's keep the conversation going. This was great. Sorry? Oh, sorry, did we have one more question? Oh, yeah, and we want to pick up Nikki's question because I think that we could have a whole session on that. I think so too. Okay. Thanks, everyone.